Hello everyone. It's my great pleasure to see you all again. In today's section, we are going to continue analysis of food products. In last video, we have seen what are the adulteration method to identify the adulterant foods. In this video, we are going to analyze the preservative methods by using the instrument. So HPLC, GC, UV, MS, PLC, CE, calorimetry, by the various type of instrument, we are going to analyze the preservative methods. The first one is what is analysis of preservatives? It is in the food and other products involves various sophisticated techniques. Here are some common methods used to identify or reduce the quantify preservatives. The first method is high performance liquid chromatography, that is HPLC. So the sample is dissolved in a solvent and passed through the column filled with a stationary phase. Different components in the sample separate based on their interactions with the stationary phase and are deducted by the deductor. After deductions, it should be passed through the data analysis and identify the compounds. Okay, so we can just see briefly how the working process of HPLC. First, the sample is prepared by dissolving suitable solvent, and sometimes it may need filter to remove any particles that could clog the HPLC column. After that, the sample is prepared. We have to inject it through the injector. The sample is injected into the HPLC system. It is typically in small volumes. That's why we are passing through the injector. Okay, typically in small volumes, maybe in the case of micromillimeters. It enters the mobile phase, which carries through the column, which enters the mobile phase and carries through the column. After the mobile phase, often a liquid solvent or a mixture of solvents flows through the column under the high pressure by giving pressure through pump. The mixture of solvent should be moved through the column. This phase moves the sample through the system. The column packed with a stationary phase, it may be usually specific chemical properties. That is where actual separation occurs. Different components in the sample interact differently with the stationary phase, causing them to travel different speeds, and thus they move through the column from the mobile to stationary column. After this, after separation, the component exits the column and pass through a deductor. After separation, the components exits from column and enter to the deductor, which identifies and quantifies each component based on characteristics like adsorptions or refractive index. Common detectors include UV visible or fluorescence and mass spectrometry detectors. We can connect through the data acquisitions. So the detector generates a signal that is processed to create a chromatograph. Peaks on the chromatogram present individual components which can be analyzed to determine concentration, purity, and identity. After the process is over, the machine will be, system is often rinsed with suitable solvents to prevent contamination and maintain the column's performance for the future runs. Okay, so this process is highly efficient for the complex mixture, and it is essential in fields like pharmaceuticals, environmental monitoring, and especially in food safety methods. So what are the applications for this HPLCs? It is widely used for analyzing preservatives like benzoic acid, sorbic acids, and parabens. So these are the main applications of that. The second instrument, that is gas chromatography. By using this GC method, how we are going to analyze the food. it should be this is the structure, this is the diagram to identify the components by GC, gas chromatography. So this is the gas flow regulator. In this way, we have to inject the sample through the inject protor. This is the detector. This will be a stationary and mobile phase columns. This will be covered with oven. The gas is it should be carries inert gas. Then after detector, it should be connected with the PC data aqua stations. The waste material should be removed. It should be recorded in the computer or data analysis. So the sample is vaporized and carried by inert gas through a column. So the sample is vaporized. Then that vaporized molecule should be added along with the inert gas and passed through the column. Components separate based on their boiling points and interactions with column material and are deducted by the detector. After the deductions, the signal should be recorded. So here they given very simple manner, but the working process is a small amount of sample. It may be gas or liquid. It may be gas or liquid. It is injected to the GC instrument, that is gas chromatography instrument, typically with the syringe. The sample is vaporized in the injection part to enter the gas phase. It's converted into the gas phase and passed through the column. This carrier gas often may be, that is inert gas, helium, nitrogen. Carrier gas should be helium, nitrogen or hydrogen. It serves as the mobile phase, carrying the vaporized sample through the system. So along the sample injector, we are passing the sample, and simultaneously, maybe in the case of carrier gases like helium, nitrogen or hydrogens, along with sample, we have to pass it through the column. This vaporized sample through the system, it flows at a controlled rate and does not interact chemically with the sample. After that, the sample flows into a long, narrow column. It will be a folded, long and narrow column, often coiled and housed within an oven. Often coiled. This column should be covered with a coiled, and it should be covered with oven. The column is coated with a stationary phase, usually liquid or polymer on a solid support. It is usually coated with a stationary phase. As the sample passes through the column, components in the sample interact differently with the stationary phase based on their chemical properties, causing them to separate as they travel through the column. So during this process, we have to maintain the temperature by oven, by increasing or decreasing the temperature. The oven temperature is carefully controlled and can be programmed to increase over a time. Temperature programming helps this. While we are increasing this temperature, this will help volatiles and less volatile compounds to separate efficiently, as each compound has an optimal temperature for elutions. A different type of samples we have, each compound will be taken here optional temperature for elution. After this, each component exits the column. It enters the stationary, it exits the column. It reaches a detector. The most common detectors for this gas chromatography, we can use the detectors in case of FID, that is flame ionization detectors and mass spectrometers. The gas chromatography mass spectrometers. The detector records a response that corresponds to the presence of each compound, what they are present. Depend upon the each compound, the detector should be record the responses. After the detector, the detector sends the signals to a computer, which creates chromatogram with peaks representing each component. Each peak denotes each component present in the sample. The area under each peak correlates to concentration of the component, allowing for the quality and quantitative analysis. After the whole process is over, the column and the injection part may be cleaned and remove any residual parts present inside the column and the injector. So ensuring we have to accuracy and longevity of the GC system purpose, we have cleaned the chromatography. Okay, it is widely applications. This GC will be mainly used for the food flavors. What are the types of flavors present in the foods and fragrance, as well as the environmental samples. At the same time, we can calculate the quality control in the pharmaceutical chemicals. It should be used for this various applications. Okay, it is the best suited for substances that are volatile. Is best suited for the volatile substance. We can identify the volatile substance by using this gas chromatography. So where it is applied, it is volatile preservatives such as sulfur dioxide and nitrates. Okay. So from the name itself, we can easily identify gas chromatography. So the sample should be injected through the gaseous form along with the carrier gas like helium, nitrogen or hydrogens. Okay. So we have to pass the stationary, we got to enter the mobile phase. So by increasing the temperature, the molecules different compound should be depend upon the melting point. It should be elutions should occur. After this, it moves through the detector. The detector should pass through the signal in the computer. Each signal should carry different compounds. Okay, so this is the gas chromatography. The next one is mass spectrometry. Mass spectrometry. Okay, what is mass spectrometry? So we already know that it should be the diagram for the mass spectrometry. So this is analytical technique. It's used to identify the composition, structure, properties of chemical compounds by measuring the mass to charge ratio of ions. Okay. So it is analytical. First when that is, it is analytical technique. Edella identify panno, which can be identified composition, structure, properties of chemical compounds. Okay. So first, see the separated components are ionized and their mass to charge ratios are measured. This is the main word, that is mass to charge ratios. That is M by Z ratios of ions are measured and providing detailed information about the molecular structure. So we can see now brief account or brief working process of mass spectrometry. First, we have to sample either in the gas, sample molecule either in the gas or liquid or solid form. It is introduced into the mass spectrometer. It is introduced into the mass spectrometer. It is usually vaporized and introduced as ions. It is usually vaporized and introduced as ions, often by an inlet system or coupled with a Fourier techniques like gas chromatography or liquid chromatography, with the help of gas chromatography or liquid chromatography. After the sample molecules are ionized in the ionization source, after the sample molecules are ionized in the ionization source, to produce as a charge particles. To produce as a charge particles. It may be in the electron ionization. It's bombarded molecules with high energy electrons, causing them to lose electrons and become positively charged by the sample vaporizer filaments. Okay. Often used large biomolecules, it forms charged droplets to evaporate, leave the ions. Okay. So once particles should be ionized, the charged particles are accelerated by an electric field. Once ionized, the charged particles are ionized by the electric field, make them move rapidly through the mass spectrometer. This acceleration enables the ions to separate based on the mass to charge ratio. Once after the ion pass through the mass analyzer, which separates them as the M by Z ratio, that is mass by charge ratio. Different types of analyzers includes, it may be quadrupole or time of flight or ion trap, whatever it may be. Okay. So by applying the magnetic field, the mass will be changed into the charge particles. This will be the mass ratio. This will be the charge ratio once it's entered the magnetic field. Okay. So after entering the magnetic field, the mass will be converted into a different range of charges of the particles. As the ion exits in the mass analyzer, they strike a detector. This will be a detector. Strike a detector after generating an electrical signal proportional to the ion abundance. Okay. So this data is converted into the mass spectrum, then showing the peaks that corresponds to the different ions. After detector, we have sent it to the signal area, that is computer. It shows the chromatogram. Each signal shows the um, peak shows the different type of ions. Okay. After that, we can mass spectrum should be value. What is the mass value? What is the charge value? We can relatively abundance of the ions. Peaks in the spectrum can be analyzed to determine the molecular weight, elemental composition, and structure of the compounds. So these are the main structures. So in this case, the sample should be a food products. Okay, the sample should be a food products. From this, we can from this food products we can calculate the sensitivity, accuracy, and complex mixtures we can identify by using this mass spectrometer. Okay. So where it is used, where it should be applicable, it is often coupled with GC or HPLC for more precise identification and quantification. We can't do, or we along with the GC or HPLC, we can identify the mass spectrometry with more precise and identifications. Okay. The next one is ultraviolet spectroscopy, UV visible. Ultraviolet visible spectroscopy. It is a technique used to analyze. This is the schematic diagram to analyze the compound. It is used to analyze the absorptions and transmission of UV visible light. The ranges we can know that 200 to 800 nanometer by the substance. Okay. This method is especially useful for determining the concentration. If you are taking the food, we have to maintain to calculate, or especially for the determine the concentration, purity, and what are the molecular structures of compounds and metals, what they are present in the foods. Okay, we can identify. Same way here also, we all very well known about this UV. So first we have to prepare the sample. It is maybe in the often liquid form. It is prepared by dissolving it in a transparent solvent. Any one of solvent, it should be. That means it should does not observe. That solvent should not observe by UV visible light in the region of interest. Okay, if you are adding the solvent, that should not observe the UV light. Then solid samples may be dissolved. If you are taking the solid samples, that should be taken in the case of dissolved conditions or specific OEDs for the gases. Okay, then we have to pass the light source. A UV visible spectrometer as a light source. Here, UV visible spectrometer as a light source. It's usually combination of deuterium lamp for UV light. For UV light, we are using the deuterium lamp or tungsten halogen lamp for visible light. The broad spectrum light passes through a monochromator to isolate the specific wavelength for the analysis. So this monochromator selects a specific wavelength of light to pass through the sample. So different regions of lights are there. It's passed through the sample. This wavelength is usually chosen based on the absorptions of peak of the compound. Peak of the compound. It is determined the preliminary tests. Okay. After that, the sample reference cuvette. Okay, so the sample solution is placed in a transparent cuvette. Okay, it should be transferred. It should be visible to the electrofluorescent. Okay, so it should be often made of quartz or glass, which allows light to pass through the sample. Okay, the reference cuvette containing only solvent is used to subtract any absorptions caused by the solvent itself. Any absorption should be caused by the solvent itself. The next one is absorptions of measurement. The selected light will pass through the dispersive element through the electrofluorescent light. The sample should be sample curved, and a detector measures the intensity of light that exits the sample. How much of peak should be exits through the detector. The difference between the initial light intensity and transmitted light in the absorptions, which relates to the concentration of the analyte. So that depend upon the concentration, the ratio will be changed. Okay. So after that, by using Beer-Lambert's law, that is data analysis, we have to undergo the data analysis. For this data analysis, we have to calculate the Beer-Lambert's law, that is A is equal to epsilon Cn. Okay, so A is nothing but absorptions, epsilon is a molar absorbivity, C is the concentration of the sample. How much of concentration we have taken? L is the path length of the light through the sample. So after this, we have to, the concentration of sample can be determined by using the Beer-Lambert's law. Okay. By using the calibration curve may also be prepared for more precious concentration measurements. Okay. So it is commonly used for determine the reducting impurities. If you are taking food particles, what are the impurities present in that food? So it's commonly used, UV visible is commonly used or identify the reducting impurities and analyzing the compounds in the system. Okay. So this is UV visible spectrometry, that is used for preservative that absorbs UV or visible light such as benzoic acids. Okay. So today we have seen 4 types of instrumentals. First one is HPLC. Second one is gas chromatography. Third one is mass spectrometry. Then fourth one is ultraviolet visible spectroscopy. Okay. We will continue in the next class. Thank you all.